This segment is segment 3 for proteins in macromolecules. And it is going to be all about enzymes. So most of your enzymes, as you can see in the picture here, are the 4th level or quaternary structure. They're pretty globular. They don't look globular when we actually take a look at what a real enzyme would look like, which I can do. I can give you one in a second. But when they're usually shown in diagrams like this, they're pretty globular. What really becomes important is the active site. And the active site for this particular enzyme in the diagram would be this stuff right in here. So this is the active site. The active site would be more appropriately termed reactive. Some kind of chemical reaction is going to happen there. In this particular case, it looks like we're taking 2 substrates. Here's one. There's another. Two substrates. Substrates are what the enzyme acts on. And we're putting them together. We get some kind of combined product here. So this was our original 2 substrates. The enzyme's job was to somehow bring them close enough so that whatever chemical reaction had to be done could be done. This type of action, a synthesis, so to speak, you would see common examples that you would, think of. Think of plants when they have to, make sugar. Whether it's fruit sugar, whether it's sugar cane. If you think of it, those organisms, those plants, they have to have enzymes that will do this, that will put these 2 monosaccharides together to make that disaccharide. When we're building phospholipids to make new cell membranes, there has to be enzymes that do this type of synthesis reaction. You will also have enzymes that can break things apart. This particular one is showing a synthesis. Let me get you an image where we show you a breakdown. Okay, in this image, it's not as fancy schmancy as the other image, but it'll do. We're starting with, there's my arrow. We're starting with our substrate here. Whoops. There we go. This we refer to as the enzyme substrate complex. You can see the enzyme action has occurred, and we get, in this case, the hydrolysis or decomp of that one substance. And so we have 2 brand new products. Okay, so just keep in mind a couple of things, because again, we're going to talk about enzymes throughout the whole rest of the year because that's how important their role is. But just a couple things to note here. The whole purpose of enzymes is that they're a catalyst. They decrease the amount of activation energy needed for the reaction to occur. So this whole reaction here would happen whenever without the enzyme. You might get lucky that they, happen to break apart. The enzyme's purpose is to allow that chemical reaction to actually occur. Okay, so without the enzyme, you may not have this reaction happen. Okay, you may not have the synthesis occur. It's the enzymes that are there that are going to be responsible for really initiating and making those different reactions occur. And I think, I know, there's one more thing I want to talk to you about in terms of enzymes. So let's clear our screen here. We need to talk about something called enzyme optimums. Whoops, undo, erase that. Enzyme optimums. Okay, so most human enzymes are going to like to function at body temperature. So one of the first things that has to be taken into consideration when you work with enzymes is temperature. Okay? Again, in humans, the optimum temperature, that's what we say, optimum temperature is going to be 98.6 degrees Fahrenheit, or it's about 37 degrees Celsius. The other thing that plays a role in enzymes functioning appropriately is pH. Whether an enzyme functions in an acid or a basic environment, acidic or basic environment, is going to play a role into how efficient the enzyme functions. So enzymes that function in acidic environments are those that function in the stomach, for example. So a lot of digestive enzymes function acidically. Some enzymes function in a basic environment. Your intestines have areas that are basic, but of course most enzymes will function in a neutral environment. Think about salivary amylase. Okay, certainly you don't want your enzymes that are going to be in your mouth, in your saliva, to work at anything other than neutral. There's a couple other things that have an impact on how well an enzyme functions, not necessarily an optimum, but we can look at the substrate concentration and I think you can probably put this together. You know, substrate greater than enzyme. It can only do so much. It's going to be pretty slow. You have enzyme greater than substrate. Pretty good reaction time, right? And so these are the main three things you typically see being involved. There are some other, different enzymes. When you have enzymes that are involved in photosynthesis, it could be sunlight that's involved. It could be that no sunlight has to be present. It could be that, water is needed. Those are the things we're going to talk about, in later chapters when we talk about enzyme function and its role when we're talking about that protein chapter. There are other things that enzymes must have to function, but we don't need to go into that just right now. One last little note, because I neglected to tell you this in one of those first videos for this protein segment. If you haven't noticed, enzymes, a lot of the time, end in ASE. Salivary amylase. Okay? You do have some enzymes that will end in IN. For example, insulin. Okay? You worked with two enzymes in lab. Oops, undo that. Let's start that again. Pectinase, yes? There's an ACE. You worked with another enzyme already. Papin. So it all depends on whether we named the protein knowing it was an enzyme, or we named it naming it as a protein. If it was named first as a protein, it probably has IN at the end. If it was named first as an enzyme, then it probably has ASE at the end. So sometimes we knew that the chemical molecule existed, we knew it was a protein, but we didn't know its function was as an enzyme. So it got named as a protein. Other times, when we know that it's an enzyme right away, we tend to name them with ASE at the end, so that we're, trying to stick to more consistent naming techniques. All right, I think that's all I have for you for enzymes now. What we do need to go into next is nucleic acids, and that shouldn't be terribly long because, as promised, whole chapter of their own. Okay, so you can move into the nucleic acid segment now.